Alright, and welcome back to Jono's lab. In today's video, I thought I'd go through the saga to date of exploring this beautiful blue fluorescing bacteria that I've isolated from some houseplant roots. So we'll start with how I found it and cultured it and figured out what it is. Then we'll look at sequencing the genome and diving into the parts of the genome responsible for this blue glowing compound, trying to figure out what that compound is. And then I'll talk a little bit about future plans. So this started by taking a swab of the roots of a jade plant that's been growing on our windowsill and growing that out on some agar. And this is just homemade agar with agar-agar powder, as well as some yeast extract, marmite, as the nutrient. And so you can see a lot of different things grew, some fungus, some bacteria. But when I shone my UV torch on it, I noticed that there was some bright glowing blue in a few spaces on the plate. So I tried to isolate that, just taking a toothpick and picking the middle of the bluest colony and streaking that out. And you can see some parts of that streak ended up blue, some didn't. There's kind of a mix of two bacteria taken over there. And so I did something called a gram stain. A gram stain is a way to distinguish between bacteria that have a single wall versus two, the so-called gram positive and gram negative. And so you can see we apply one stain, then we apply iodine, and then we wash it out with alcohol, and then we apply a second stain. And so the Pseudomonas here is this light pink. It is a gram-negative bacteria, and so they're taking the first stain, but then they have just one thinner wall, so that washes out. Whereas these other little larger round dots, cocci, as anything spherical in bacterial world is a cocci, those are the sort of contaminant in our plate, so some other soil-dwelling microbe. Anyway, so it took me a couple of restreaks of just picking, just trying to find just the edge where it was mostly blue, and then streaking that out so you get lots of isolated colonies, and then picking the ones that were blue. A couple of restreaks I managed to completely isolate just the Pseudomonas, or at least at that stage I just had a guess that it was a Pseudomonas. They're known to make these sort of blue glowing compounds. Okay, so one next piece of like plausible identification was to see whether, like, the literature says these pyovadines bind to iron. So seeing if binding to iron changed the fluorescence, which it does in pyovadine. And so I ran a little experiment, just putting two identical pieces of liquid that were filtered so that there were no cells remaining, just the stuff that had been secreted, and it glowed beautifully under UV. And then putting in one of them water and in one of them some vinegar that had been sitting with some rusty wire inside it and a little bit of citric acid just to dissolve a bit of iron. And as soon as the iron in solution hit the pyovadine solution, the glow instantly went away, and you've got this sort of dark cloud of precipitate there. So I think I'll insert a video of this happening as well, and I'm going to try and get some better footage in the near future. Okay, so this is cool. We have a sort of plausible idea for who this is. I did some agar art with it. The green and the red here are modified E. coli that are expressing fluorescent proteins, but this blue here is just the pyovadine itself, no modifications needed. Sorry, the Pseudomonas itself, because it secretes this glowing molecule. Okay, and then I've had some fun making large amounts of this by growing it in liquid culture, so just 500 mils of broth, and leaving that for a few days for the bacteria to multiply, and then doing something called spherification or reverse spherification to turn that into these gelatinous blobs, or in this case, the sort of gelatinous goo. And I noticed so obviously being somewhat careful because it's a living thing, although these are usually not going to attack you unless you have like a weakened immune system and they somehow get inside. But even after pouring out the gelatinous goo and then sterilizing with boiling water and then spraying with alcohol and then handling it to wash up, I noticed that even after all that, there was still enough of the pyovadine that some of it stained my skin, which wasn't visible except under UV. Oh, that's actually kind of cool. I think that'd be fun to try and like figure out that process. But at this point, I was starting to think, well, if I'm going to be doing this, if I'm going to be wanting to handle this stuff, maybe if I think it is a beneficial soil bacteria and I want to be spreading it around my garden or starting to make cool skin dyes or any kinds of further experiments, I should probably know exactly what it is. And I should probably check, A, that it's safe, and then B, can we figure out exactly what this molecule is? Because pyovadine is not one molecule, there's a whole host, hundreds of different forms, right? So they all have some pieces in common. The chromophore is generally the same, but there's different side chains, different peptide chains within the molecule that give each kind of variant its own characteristic structure and performance as well. Okay, so it'd be really cool to figure out what that is. So how on earth do you go about doing that? Well, I have my isolated bacteria, and so what I was able to do is take a small sample, just 10 milligrams, 50 milligrams of cells in a liquid to kill and preserve them and preserve the DNA. Ship that off to someone called Plasmidsaurus, who is a fantastic company. They have an office here in Eugene in Oregon. And so I shipped them off, I think, on Wednesday morning, and by Thursday afternoon, they had finished sequencing the genome, and they were able to send me a whole dump of data, including the raw genome reads, as well as some annotations. And so they give you a nice little report that says, hey, this is the number of reads. They have different services, but I knew most Pseudomonas species tend to be around 6 to 7 megabase pairs in the genome. And so that I just chose their sort of minimum bacterial genome service. I think it says it handles up to 7 MB fine. Yeah, so we get this cool analysis. One of the things they tell you right off the bat is that they do 2 different ways of saying, hey, which is the sort of nearest known genome to this? And you can see here both of them have responded with Pseudomonas putida. My apologies to Spanish speakers. So this putida it's not exactly a species. In bacteria, species are more complicated than you may be familiar with from other parts of the the domain of life. So this is been identified and categorized, but it's kind of a they call it a group, the P. putida group, because there's many different strains, including many different variants of pyovidine produced. But this group as a whole, it's a soil bacterium. It tends to, because this molecule it secretes binds to iron, it tends to be useful for plants and other bacteria around to be able to uptake iron if there's Pseudomonas also present. But it's also kind of used for the like interbacteria chemical warfare that's always going on. So it can be like it's a defense mechanism as well. But anyway, kind of cool to see, and this is a group that's being studied. There's various work being done, kind of splitting it up based on what variant of pyovidine it produces. Okay, so we've got like a kind of like a ballpark, and the other thing that's lovely about this is that this is host vector system safety level 1. So this is safe to use. It's not like pathogenic. Some of the other Pseudomonas species, it's not like they're like active pathogens of humans. But if they get on your, if you're getting a surgery or something like that, and some gets in, and you have a weak immune system, there can be some like worries of infection, especially with this particular variant, for example, is kind of more studied because it has had some interaction with people, the opportunistic pathogen, they call it. Whereas P. putida, sort of known, safe, much less of chance of that happening. So still, obviously, I take all the care that I usually do in the lab, but it's nice to know that this is a fairly benign host. And this is good because I'm also planning on having this be kind of a model organism that I can work with. It smells much less bad than E. coli. It grows very readily on any media that I seem to make. Its glow makes it very easy to identify that you haven't got contamination. Many, many benefits. So I think it's kind of cool to start knowing what it is. Okay, so then the big question, how on earth do we figure out what exact molecule that it makes, right? Because like I said, there's all these different variants. So this is where we get into the idea of genome mining or analysis. So I ran a lot of the work in something called Solver, just like an interactive notebook. So step 1 is loading up the annotation, the big list of basically what Bacter gives you is a list of pieces of the DNA that they have sort of a reasonable guess as to what that chunk might be, right? This might be some particular known protein that is similar to something that's already been studied or categorized, and so they'll kind of tell you, look, hey, here's a list of all the locations in the genes, like where it starts, where it stops, what we think that might do. And so if we go and search for pyovidine-related genes, you can see that we have a number of hits. And so the way a molecule like this typically ends up working, for one thing, they tend to cluster together. And the reason is that when you've got a secondary metabolite, a lot of the intermediate products might be somewhat toxic, but also they're not really useful on their own, right? So if you had all the genes for this spread out across the genome kind of randomly, odds are with a lot of the recombination and cutting out and horizontal gene exchange, it's unlikely that 2/3 of the genes, if they were transferred, would be useful at all, right? Because if you only cut out one section of DNA, you're not going to get everything needed if it's all spread out. So what this tends to select for is for functional units, i.e. all of the pieces related to this molecule will tend to cluster together. And so we call these biosynthetic gene clusters, BGCs. And I'm going to get a lot of I'm going to say the things wrong and pronounce them wrong because this is all, I'm somewhat of a noob and I never have heard other people say any of these things. So if my pronunciation and acronyms are slightly off, you might want to double check this. But anyway, so we're looking for a cluster of genes related to pyovadine. And you can see that there's a lot of these ones starting with PV. These are like known genes for the pyovadine system. They're all together in this section along with these NRPS genes. Okay, so starting to like zero in of this whole long genome, there's one section that seems like it has most of the pieces. And so this BGC is the thing we're going to dive into and expand and see what's going on in there. And so you can see pyovadine export, right? This one in particular is responsible for secreting the molecule once it's made. PVD blank like protein. This is the best the annotation system can do, but we can start to dive in and understand a lot of these. And so then there's a few interesting ones. This one here is a large non-ribosomal peptide synthetase, and they've labeled it Sol-G, which is Sol-Anamycin. But I think this might just be a mislabeling, and you'll see why. But then we've also got another one here, for interacting fat binding protein. But you can see just like if you've been reading around pyovadines and siderophores, this seems like the cluster that we're interested in. And specifically these two big non-ribosomal peptide synthase molecules. These are like factories where they're going to have a number of sites, binding sites, A sites, adenylation sites, I believe they're called, where it's going to be like a pocket that particularly attracts just one specific amino acid, and kind of brings it in, uses some ATP as energy to process it and add it to the chain or whatever, and then pass it on to the next A site for the next one. So this is like a little factory that without needing the ribosome and the normal protein synthesis pathway, this is just going to make a short peptide chain. And then there's going to be various other ones that are going to do some modification to maybe some of those pieces. Okay, so we'd like to like analyze and dig into these things. So the next tool in our toolbox is something called Anti-Smash, and this has a web user interface, so you can upload the data that you have, and it's going to try and identify these BGCs and annotate them. And so that's what I did. I uploaded my sequence, and it identifies 10 different regions, and specifically the 10th one is the one that I think is interesting because this is where we have this kind of like lines very neatly with our identified cluster, right? The same sort of region of the genome. And we get this lovely little explorer. You can see for each of these ones, you can go and look at like, oh, I'd like to look at, for this one specifically, could we look for similar ones? And you can see some of the top results, the sort of most similar pieces. It's very useful to start to piece together like what other known variants this is most similar to. But also it gives us these predictions for what the A domains are. So these are not always perfect, but if we run this on our region 10, that one that looks like it has all the pyovadine related pieces, we get this list of modules. And it's very exciting because if you look at the structure of pyovadine, they all start with this conserved dihydroxyquinoline core, right? And so this is built from, in the precursor, this is built from, they listed here as Q because it's always the same, it's always invariant, and it's always glu-tyre-dab, right? And so then this is going to turn into one of two different side chains. These two are going to be cyclicized into that dihydroxyquinoline module. But the fact that we have here a sequence that starts with the known chromatophore core is a very strong indicator that this is the one that was labeled as Sol-G, but is actually probably our pyovadine key NPRS. And then this gives us a reasonable guess for what the remaining pieces are, right? So we have ASP, ALA-ASP, ornithine, and then X is unknown, and another ornithine. And this X is because sometimes some residues are harder to predict than others, and it's not obvious, it's not close enough to some known thing, or it doesn't match anything in the library quite perfectly. It's not obvious what this particular residue is. But that's okay. We've got a reasonable guess. These E-domain actives tell you whether it's L-glutamine or D-glutamine, so you've got the chirality orientation, I forget the correct term for that. But anyway, so we've got a reasonable guess as to what our molecule looks like, right? At least this part of the precursor. Okay, so that's exciting. I did various bits of work trying to look at ways of determining what this mystery residue might be, and had different guesses at different times. But I'll come back to determining that. So there's a few remaining questions still. This is just the sort of core peptide backbone of the precursor molecule. Like I said, these ones are going to get oxidized and cyclized together into the sort of final chromophore. This one's going to get a side chain added, so that's something we should look at. And then often ornithine is going to get formulated into a slightly different form. So we can start to dig into that. There's known genes that will turn that GLY into one of the two sort of common side chains. Some pseudomonads have both. So I found reference sequences for both of those, and BLAST just looking for like alignment and similarity. I guess you get a kind of similarity score, and you can see we got a fairly high similarity to PBDN, fairly low to PTAA, which is kind of a reasonable hint that probably we're producing the succinimide sidechain variant only. But obviously, like, there's some that are slightly different. That's still to be determined. But so that's one mostly question answered. And then this mystery residue, I'll show you what ended up happening. So obviously, like when I started to see the predicted sequence, I did various searches of like PubMed and Google Scholar, and I also had AI searching around for this string of amino acids with no luck. But then at some point I was browsing this paper on Sci-Hub, and I'd come to this before because it has this nice table. It's talking about P. putida. It's talking about it's like giving a bunch of different pyridine, peptide chains, and I skimmed, but I'd like stopped about here. So then I came back to it and I scrolled a little more and I saw this, which is pretty much bang on for ours, right? Asp, ala asp, ornithine, serine, ornithine. And that is an exact match for ours with the serine in the position of our mystery residue X. And ser is hard to predict. And I guess the reason that I didn't catch it is because just this notation for I think I wrote it down somewhere here. Let me just get my pronunciation and words correct. Yeah, amide-linked hydroxyornithine versus cyclic hydroxyornithine. So they just used a notation, a very useful one, to tell us that these hydroxyornithine molecules are what they look like in the structure. But it meant that the exact string matching wasn't finding that. So little lesson there, actually reading the papers yourself with your eyes, and fuzzy pattern matching as a human can sometimes still be the best result. Okay, so we've got a reasonable guess for the serine, and then also the other part was, like I said, sometimes these groups are formulated, and so there's a gene called PVDF, right, that's responsible for the protein that does that formulation, and I couldn't find that in our biosynthetic gene cluster, and so yeah, the fact that we found a P. putida species that has a sequence that's a very close match to ours, that has unformulated hydroxyornithine, and that has a somewhat hard to predict residue exactly where we had our mystery one. This gives us a pretty strong confidence that this is this is sort of a close match for our molecule. I still am hoping to actually take purify it and run something called liquid chromatography and then mass spectrometry to like analyze the molecule and see whether this is the right guess, whether we do have the same as the string, because there's always a chance that this is some different residue, right? It's mutated and found some difference, and also that our guess on the side chain is right. But it's pretty cool because this lets us then take a look at the molecule. So this is the precursor. This is before these two cyclize into the core, before this gets turned into the side chain. And this serine here is our best guess based on that close match. But it's pretty fun to be able to see the molecule, to be able to plot it in 3D. So this is the structure, um, at least a better guess as to the structure. And so you can see these are going to bind together. The whole thing might coil up a little bit. But it's pretty cool because yeah, I don't know, this gives me a much more tangible sense of like what is being produced. I feel like it's fun to start digging in and answering this mystery. And these hydroxyornithine groups, these are both possibly going to be binding to iron 3 plus as well as the chromophore core. So it's a very strong iron binding molecule. Um, Pretty cool. And wouldn't it be fun if we could purify this, isolate it, verify it with the mass spectrometry, and then start to see what other fun things we can do with it. Maybe if we can turn it into the different forms that fluoresce in different colors by modifying the molecule somehow. Um, yeah, okay, so that's the process. At the moment, I'm just having fun. I've got a CNC pipette. I've been using this as a paint to make some fun little pixel art. Yeah, been messing around, growing it out and trying to purify the pyovadine. But yeah, I just thought I'd make this long video. Sorry if it's been a little rambly, but to basically take you along on that journey of diving in and to show that it is possible to take some unknown organism from the world, right? Um, Sequence it, see that it's maybe especially with bacteria, it's always the case that in the analysis, it's only a 95% match. In other words, there's always going to be differences. The one you have is always going to have some genetic drift from even the nearest sort of known neighbor. And then be able to dig in and say, okay, like let's look at the genome, let's see the kinds of compounds it's making, right? In this case, I have a particular interest in this fluorescent one, but there's no reason you couldn't go through like looking at some of the other NRPSs, or just poking around and seeing what other annotated genes there are in the genome that could be interesting. It could be fun to then try, like, for example, using CRISPR to knock out one of the genes in this pathway and see what happens there. Yeah, and basically start to explore at this very granular level what this little tiny creature that you can't even see with your eyes is doing and what kinds of things it's producing. And this, like the chemistry of making something like this is wild, right? Like to try and make something like this in a lab is going to be incredibly difficult and expensive and complicated. And this biological machine just creates these proteins that pop this all together. So anyway, I find this fascinating. I hope you do too. Stick around for future adventures on this if I manage to do the mass spectrometry. But for now, thank you so much for watching, and we'll see you in the next one.